So let's get started. I think it's really important that I talk a little bit about the collaboration for change and how I got involved with CVI. So I am, as you know, the director of the Laboratory for Visual Neuroplasticity, the Mass Eye and Ear in Harvard Medical School, and I have the pleasure of working with some really fantastic individuals at the lab. Karina Bauer, who I'm sure many of you know, who's assistant professor and now actually leading her own independent line of research. Zahida Pamir is a postdoctoral fellow and will be moving back to Turkey, where she's going to be starting her own lab doing developmental research as well. Claire Manley, say hi, Claire. Claire Manley is the study coordinator for the study. Many of you have already interacted with her. We have Chris Bennett and Xin Zhang, who are no longer with us, but really played instrumental roles in developing a lot of the strategies, a lot of the tasks that we work with as well, and I wanted to acknowledge them. The history of the lab was really, at the beginning, about ocular blindness and how the brain adapted to ocular blindness. So then I would go to these various meetings like AR and so on, and I would meet people like this person. This is Peg Palmer, who many of you know. She's a teacher for the visually impaired from Connecticut, and Peg was in my ear for about 15 years about CVI. She would come to all my lectures, and at the end of the lecture, she was like, what about CVI? What about CVI? You need to work in CVI. CVI is really important. CVI is really important. You need to work about CVI. And I would say, Peg, what's CVI? I had absolutely no idea at the time what she was talking about. And the more I thought about it, the more I sort of dug into it, I realized that this was an absolutely crucial direction that we needed to think about. So one of the very first conversations I had, you see Dr. Barry Cran, who is here, Dr. Louisa Meyer, and as well as Derek Wright. I call them the three musketeers of CVI. And one of the very first conversations I had with them was to try to understand what were the challenges that needed to be addressed, what were the gaps. And one of the biggest gaps that were identified from our conversations was the importance of assessment, having objective ways, and of course, Glenn's talk is exactly was along that line. The next person I talked to was Peter Becks. He's a professor from Northeastern University, and like Glenn, loves to test vision. That's what they do, and coming up with new assessments, new tasks. We've been collaborating with him, and I'll show you some of that work. And then finally, Dr. Jenna Hadari, who will be giving a virtual lecture after this. She is a pediatric neuro-ophthalmologist. Again, pediatric neuro-ophthalmologist. Very handy skills if you want to work with CVI, obviously. And she was very, very helpful as well in those initial dialogues to understand CVI, particularly the eye versus brain component, right? And that's pretty much how it started. Then came the collaboration with Perkins. And I remember Ed Basso, who is here. I met Ed about 6 years ago at an event at Mass Eye and Ear. He came up to me. He said, hi, my name is Ed Basso. I'm the new superintendent of the Perkins School for the Blind. We're doing great stuff on CVI. You should join us. You know, I'll reach out. It's going to be really, really great. And I said, I will never hear from this guy again. I couldn't have been more wrong, and I couldn't have been happier for that reason. Ed was actually worse than Peg. This guy was emailing me every day, where are you? I know where you work. You got to come. You got to collaborate with us. And of course, with his leadership and the team that he put together at the CVI Center and the wonderful staff that you see working here today, this is where we're at. So it's his leadership and this incredible team that he put together, and I'm really, really grateful for that opportunity to work with them. And I honestly believe it's the best thing that ever happened to the project, because not only did it give us a sense of purpose, it gave us a sense of community. And that is the most important thing, I think, that came out of all of this. So I'm really, really grateful for these collaborations. You can see the list is growing, right? Not just regionally and locally here, but also nationally and internationally with Italy. Other collaborators, again, Emory University, the Gordon Center for Medical Imaging, Northeastern, as I mentioned, Italy as well. So it's growing, it's growing, it's growing. And then finally, I want to mention our funding support. We started off with very, very small foundations who believed in us, who believed in what we were trying to do, but weren't so hard necessarily on the data just yet. So in our early days, we were just figuring things out, and we've been very, very fortunate to receive substantial funding from the National Eye Institute since that time. So it was a long journey. So I love this statement. It's not the beginning of the end, but it's definitely the end of the beginning, and I think it's really been a wonderful, wonderful journey. So I'm now really anxious to share some of the results that we've been working on. This is a seminal paper. It was published in 2007 by Dr. Elisa Fazzi, who's in Brescia, Italy. I consider her the grandmother of CVI, and one of the very first people thinking about CVI before we even called it CVI. really a seminal piece of paper, like I said, published in the Journal of Child Neurology in 2007. She looked at 121 patients with visual impairment associated with damage to the brain, roughly 50-50 distribution, about children born term and those born preterm, roughly mean age about 54 months, about 4 or 5 years old. And the first thing she noticed in these 121 patients was the cause associated with their visual impairment. What you see here highlighted on the top line is hypoxic ischemic encephalopathy, so a stroke, if you will, right, associated to happen during development. But you can also recognize that there are many other causes as well, genetic disorders, metabolic disorders, epilepsy, and that really first gave us a sense that there's not only CVI, but there are many causes of CVI as well, which makes this very, very challenging. She then went through and systematically looked at all the various aspects of vision. So you can see this broad breakdown here. So many of the kids had refractive error, many had ocular nerve issues, many had strabismus, many had reduced acuity. But the most striking thing that I found that came out of this report is that almost 90% of the kids had impaired visual perceptual abilities. The most common thing that they had were perceptual issues. And at the time, it was assessed using the developmental test for visual perception, which many of you, I'm sure, are familiar with. Very, very useful, but it was never designed for CVI, right? So if that child or if that individual shows some deficits or some issues, why? Did they understand the task? Did they have problems using the pencil? Did they have sufficient visual acuity to even to see the targets? Were there clutter issues in order to sort of somehow change the way that they perceived or make it a little bit more challenging? So that's something that always kind of stayed in the back of my mind. And like Glenn said, we need better ways to do things specifically for individuals with CVI. There's a great sort of hierarchy model that's coming out of the Netherlands. They talk about lower order issues in CVI, so sensory visual issues, ocular motor issues that Glenn was alluding to, and the second tier, what we call higher order, which are really more these perceptual functions, visual orienting, attention, visual spatial processing, visual identification. And I can tell you that our group is largely interested, or concentrating, I should say, on the latter. That's not to say that this is more or less important or anything along those lines, but as you saw, CVI has a very, very broad presentation, and this is largely the aspect of CVI that we're concentrating on. So just a quick summary here. So CVI, cerebral cortical visual impairment, assessing higher order processing deficits. As you know, there's often the presence of unique higher order visual processing deficits, visual spatial issues, complex motion processing, visual search and attention. And Gordon Dutton typically is associated with this term. He calls this dorsal stream dysfunction. So just to remind you, there is a useful conceptualization of the brain from the occipital cortex, the parietal cortex, to the frontal cortex. That's the dorsal processing stream. That's the where pathway, or vision for action. The ventral stream from occipital cortex down temporal cortex, that is the what pathway. That's your visual archive, right? That's how you are able to identify things. I completely agree with what Marguerite said yesterday. Very useful way to think about visual processing, this division of labor, but it does not operate in isolation. The brain is constantly exchanging that information, right? So again, useful conceptually, but it's really important to think about the brain holistically, right? It's constantly communicating, and again, show you some evidence about that as well. So here are the things that we typically hear, right, about kids with CVI. So a great little summary slide from CVI Scotland. Sometimes I can't see objects in the distance, especially if it's busy or cluttered. I sometimes can't find a person I'm looking for in a group, right? We have that classic scenario, for example, you show a child their favorite toy and they say, yeah, this is my favorite toy. You put it in a toy box and they can't find it anymore, right? Then they take the toy box and then we call them dumpers, right? And they spread all the toys out and try to find the toy box. What is that? That's not just visual acuity. There's much more going on here, right? The problem is, as Glenn very elegantly presented us, it's very hard to figure things out when you're starting with an eye chart, right? There's a huge gap between what they're describing and what you can draw from just looking at their visual acuity. What we really need is a measure of their functional vision, how they use their vision in the real world, and that's the gap. And we think the best way, or one of the best ways to close that gap is virtual reality. So that's the big sort of direction that we are heading in, right? So very, very similar in terms of what Glenn is doing, new tools, new way, and we are concentrating largely on these higher order perceptual issues. So why is virtual reality useful? One is you can create the environment, right? You have control. You can create the level of difficulty or sophistication that you want. And coupled with eye tracking, much again like how Glenn presented, you now have an objective measure. You're not reliant on a verbal response response or a motor response. You can create it at a sufficient level of visual acuity that the child or the individual can interact with, and you now have an objective way that avoids a lot of these confounds with classic neuropsychological testing. So that's the point of this approach. Why do we use eye tracking? Just again to add on Glenn's presentation, eye tracking is extremely helpful in telling you what that person is thinking or what that person or how they are perceiving the visual world. This is a classic study by Yarbus, one of the first people to document eye movements and how they're in relation to visual perception. So you can see that eye movements are really, really kind of chaotic looking, but they're not random. Right? There's a sense of what those eye movements do. So for example, when we look at a face, we don't just look anywhere on the face. We spend a lot of time on the eyes, the nose, the mouth, the outside of the face. So the brain is picking or telling the eyes where to look. Right? It's strategically using information and drawing that information out of the visual scene. So where the eyes are looking tells me something about how you perceive the visual world. The second thing that's important to think about is visual search as a task is extremely demanding on the brain. It's not just the eyes, it's deep structures in the brainstem, it's subcortical structures, it's the visual cortex, it's the dorsal stream, it's the ventral stream, it's frontal cortex for attention. So you might imagine that any trauma to the brain, early or later in life, is probably going to manifest at the level of visual search. And that's why it's so useful, I think, and so sensitive, is because it requires so much brain power to do it, right? So what we did is we conducted a focus study. We brought in people who we thought would be the most informative. We didn't think that we knew what the best task was. We actually sought out input. So we brought in potential participants, parents, teachers, individuals, as I said, and conducted a focus group questionnaire to try to figure out what were the best environments we should come up with. And there were two that came out. One was finding a toy in a toy box, right, identifying an object in scenes of clutter. And the second was identifying a familiar person in a crowd. Right? So this was more an issue of crowd density, right? So those were the two scenarios that we were interested in. We then took that information and converted it into two virtual reality environments. I'm going to show you the data from that work now. Again, many of you have seen this. I promise you, I do have updated data to show you. So we have now almost 60 participants who have gone through the toy box. These are matched by age, and I'll have a comment about that related to what Barbara said. On the left is an individual who is a control, and on the right is an individual to the right. But before I show you this video, the toy box is actually quite simple. It has 3 levels of difficulty: low, medium, and high. You'll notice there's always 25 objects presented there, but what we change is the number of unique distractors. So this is a task that's defined by clutter. The harder it is, the more unique distractors that are present, we think the harder it is to find the actual target, right? So that's the issue of task difficulty. The first thing they have to do is identify the toy that they want to search for, so it's engaging, and we know that the child can identify the toy in isolation, then put it in this little 5x5 matrix, and then we track the eyes, right? So that's the idea. So again, on the left is a control, on the right is an individual with CVI. That yellow blob in the middle there, that's where their eyes are. And I've slowed this down to about 75%, so you can see where the eyes are going, all right? So what I want you to concentrate on is notice on the control how the eyes move versus the CVI participant, and the task here is to simply look at the blue truck. And the first thing you should probably notice is that you notice how for the CVI individual, that path is much more complicated, right? So think about that. If I ask you to walk from this podium to the back of the room straight, not a problem. But if I then ask you to take this long, long route through all the tables, you'll get there, but you're going to be tired really, really quickly, right? And we think this is a reason or part of their explanation as to why children are so exhausted at the end of the day. Their eyes work so much harder to do exactly the same thing. And again, this is not something like Glenn said that you would pick by just looking at my finger, right, in a standard neuro-ophthalmology exam. You need the subjective quantification in order to see this phenomenon. And here's what the data looks like. So I'm plotting success rate, the likelihood of you being able to find the target. In blue is our controls, in orange is our CVI participant, and our CVI participants, I should say. And the success rate is this. It's about 98% in our controls, 71% in our CVI participants, right? You can see that overall they're less likely to find the actual target. But here's the most interesting thing. This is what's called a box plot. This is the distribution of the data, right? So the box plot is the range that you see. The individual circles are individual participants. So while the controls are super tight, they're all kind of behaving the same way, look at the range that you see in CVI. Again, something you all know. There's a massive variability when it comes to CVI. And these are all individuals who are all category 3 or phase 3, you're all familiar with, right? These are, quote, high-functioning individuals, right? Look at the variability just in that. In fact, our best CVI participants are doing better than our worst controls. I'm going to repeat that. Our best CVI participants are doing better than our worst controls. Those are the kids who fall through the gaps, the cracks, right? So that's why the overlap, the fact that there is an overlap is extremely crucial to kind of think about, right? We'll go even further than that. Here is the same data, but now I've separated the CVI group based on entering visual acuity, right? So the controls in blue are there. See, the arrow is a little bit lower for those CVI participants who have 20/20 or 20/25 visual acuity versus those participants on the right, the extreme right there in red, who have 20/30 or above. And you notice that CVI participants with normal or near normal visual acuity are performing about the same rate as those who have reduced visual acuity. So in other words, good visual acuity does not make this go away. This drives home the point that this is a brain-based issue. This is not an eye-based issue, right? Last piece of data I want to share with you with this particular slide. This is reaction time as a function of task demand. So the higher you are on this y-axis, the slower your response was on the task. On the x-axis, moving to the right means the task is getting harder. So low, medium, high. This is what it looks like in blue in our controls. It's pretty much flat. So in other words, as we change the number of unique distractors, it does not affect their reaction time. But in CVI, it certainly does. Not only are they slower overall, but as the task gets harder, their reaction time gets longer. And we call this a load effect. And this, we think, is related to this idea of complexity. As the task gets harder, as the task gets more complex based on clutter, their reaction time starts getting longer, right? And that load effect, we think, is really a cardinal sign of what CVI represents. We've also coupled the toy box now, this is new data, with EEG, electroencephalography. This records brainwave activity in the brain, right? So now we're recording using this EEG device with the toy box. So the individual is doing the toy box task. We're recording not only the eye movements, but also now the brain activity. And the nice thing about EEG is that it tells you when things happen in the brain. It has excellent temporal resolution, right? It's also useful because you can break down the EEG signal in all these frequencies, what we call theta, alpha, beta, and gamma. Alpha is really a strong signal in the brain when it comes to visual processing. So when you open your eyes, the back of the brain has something what's called desynchronization. So it's the visual cortex firing like crazy because of that complex visual scene that it's trying to analyze, right? So I'm showing you here in the upper right in controls, this is frequency as a function of time. The dashed line represents once the toy box appears, and then we record brainwave activity afterwards. Do you see this blue little target area here in the middle? This is alpha desynchronization. This is the visual cortex coming online and firing like crazy once the toy box appears, right? Classic presentation in terms of how the visual cortex normally fires, this alpha desynchronization signal. Here it is in CVI, virtually absent. There's actually, I remember my collaborator, David Batari, when we sent him the data, he goes, there's something wrong with the data. He goes, this doesn't make sense. I've never seen this much alteration in alpha desynchronization signal. You can see in CVI, not only is the alpha desynchronization signal absent, the other frequency bands are out of sync. And it's interesting because we think alpha desynchronization is like a clocking mechanism, right? It helps the visual cortex synchronize the brain. And because that alpha desynchronization isn't there, the rest of the brain is not locked into it. Could that explain visual latency, for example? So I want to be very, very clear. It does not mean that the visual cortex isn't firing. It is just not synchronized in a way that we normally see in neurotypical development. The second task I want to share with you is the hallway. Again, you've seen this, and I'll show you some more comprehensive data from this. Very, very similar to the toy box, except now we're adding movement. So as Glenn said, it's not just a saccade to the target. You've got to search for the target, find it, and pursue it. So now we've added this extra layer of complexity. Very, very similar. 3 levels of difficulty, low, medium, and high. Here you have to choose a principle based on race and gender matches. You choose the principle you want to look for. The principle is in a crowd, walking, and we change the number of individuals in the crowd. So on the left is a control. On the right is an individual with CVI. This person is searching for the white female principle. You'll see the control finds it on the right there and pursues. You see the delay for the CVI individual. Show you another example. Here the control is searching, locks in, finds them. CVI individual still looking, right? So an extra layer of complexity because of the motion. And we think, similar to the toy box, as the crowd gets bigger, it should be harder and harder to find the target. And indeed, that's what the data suggests, right? And again, there's that big distribution that we see in CVI that we don't see in controls, and that load effect is there as well, right? You can see that as the task gets harder, their reaction time gets slower. We've now coupled this with fMRI, because this is a task that's actually very, very good for functional magnetic resonance imaging to see where things are happening in the brain. So EEG is very good for when things happen. fMRI is very good to figure out where things happen in the brain, right? So here's his group data. What I'm showing you here, first of all, this is the right hemisphere of the brain. This is the occipital cortex, parietal cortex, temporal cortex. The eye would be somewhere here in the front. And the first thing to take away from this is that when participants are in the scanner and they see this task, it drives the visual brain, right? Occipital areas are active, parietal areas, frontal areas. It's hard to see, but if you turn the brain on its side, you'd see the temporal stream, the temporal cortex active as well. So it's a strong, strong task to activate the brain. What this doesn't tell you is how strong any particular area is active, right? And to do that, we do a region of interest analysis. We look in particular, how strong the response is. So here's the behavioral response that I told you about. There's that load effect in CVI that as the task gets harder, the reaction time gets slower. And this is what it looks like inside the brain in terms of the overall response. So this is early visual areas, V1, V2, V3, the first levels of visual processing. Again, orange is CVI, blue is controls. And do you notice how there is this load effect, just similarly in terms of brain activation, just like there is in the behavior? As the task gets harder, the brain is working harder. But the other thing to notice is that there's almost perfect overlap, right, between CVI and controls. So that means that whatever differences we see between the two groups, it's not happening at the level of these early visual areas. So there's somewhere else that must have to explain for this. So if you march along the dorsal stream, now we start seeing separation, right? So you notice how in every single one of these areas, the dorsal, the blue controls is always higher than the orange CVI. This seems to line up with dorsal stream dysfunction, right? So the dorsal areas are not activating as strongly in CVI as they are in controls. It makes sense. What about the ventral stream? Might as well look, we're here, right? First area we looked into, this particular called VO, exactly the same thing. Looks like business as usual, right? Controls firing much higher than CVI. But then we kept going and something interesting happened. It flipped. Now CVI is firing harder than controls. So now this begs the question, in the context of dorsal stream dysfunction, is there ventral stream compensation? And our initial data suggests that that's actually true. I have some suggestions to think that this is actually going on. If you now correlate behavior with activation, it seems that this may be an explanation. So I think we can now take this hypothesis even further. So it's not just dorsal stream dysfunction, there could also be ventral stream compensation. Last task I want to share with you, this was a collaboration with Matt Tejan. Okay. Perfect. Slight, before I add, Mary tells me we're going to ask the hotel to delay the checkout, so not to worry. All right, we're still doing okay? All right, perfect. Thank you, Mary. Last task to share with you. This is something called the 2D image study that we did with Matt Tejan, who you're all familiar with, right? So he's a teacher for the visually impaired in Connecticut. Emily Cantion, a teacher from Perkins, and a collaborator from Italy named Serena Mecaletti. Matt came to us and said, you know, I noticed in the classroom that a lot of my kids really struggle to look at things like cartoons and comic books. They just don't quite understand the images, right? But if I show them real pictures of the same thing, they get it. What's that disconnect? What's going on? So we had this conversation with Matt. He came to the lab, and he shared with us a bunch of pictures of 12 common objects, right? A cat, a dog, a car, etc., with real pictures. And then we made different versions of those images. So an abstract outline of a cat, a realistic outline of a cat, an abstract colored version of the cat, and a realistic colored version of the cat, and then finally the real photo itself. We take all these images, so 12 by 5, that's 60 images, we scramble them, we put them on a laptop computer, there's Claire doing the task for us there, and we simply ask them, tell us what you see, just name the object. And when you name the object, press the space bar. Right? So that also gives us a reaction time to see how fast they're actually doing. So it actually gives us a chance to disentangle what is the driver here. Is it color or is it the abstract nature of the image, right? Then Matt said, you know, it would be really nice to know where they're looking when they do their task, right? I said, sure, we can do that because we actually have an eye tracker, right? So we have it hooked up. So when they see the image, as soon as the image appears, we record their eye movements until they give us their response. So we have exactly their gaze on every time they identify the object. So you can see in controls here, it's really, really tight, right? In CVI, looks a lot more scattered, right? So is there any relationship between where they're looking and what the actual image is? And to do that, we use a technique called GBVS, right? This is graph-based visual saliency. It's a mathematical way to represent the saliency of the image based on luminance, based on color, based on the proximity of edges. And you can see when the mathematical equation comes out, you can see things that are particularly salient are things like the face of the cat, the tail of the cat, right? The legs of the cat. So we now have an objective measure for the saliency part of the images. So then what we do is we actually compare the two. We take the saliency map of the image, we take the person's gaze, where they looked, and we see whether or not there's a correspondence between the two. And mathematically, the way we do this, thank you, Mary, looks like we're good for the delay. And the way that we do this is do something called an ROC analysis, or receiver operating curve. And the idea here is the higher the value, the better the correspondence between the two, the prediction and where they actually looked. And here's what the data looks like. So we have now over almost 90 participants in this study. This is a collaboration with 80, and I can tell you that when you've got 90 participants, you start believing what you see. So the power of collaboration for sure. This is percent correct in our controls as a function of the image type. So abstract black and white all the way to realistic photos. And our controls in blue are at ceiling. They can identify all the objects, no problem. Here's our CVI participants. Isn't it interesting that as the image gets more and more add information linearly, they actually are able to more and more identify the actual object as well. So our abstract black and white is the hardest. When you add a realism to it, they do better. When you then add color to it, they do a little bit better. And then you add the two, they're doing a little bit better all the way up to realistic photos, right? So it makes sense, objective way to understand what each other contributions are for image identification. Reaction time, we see this nice trend in both groups that as the image goes from abstract black and white all the way to color photo, the reaction times get faster. And the last thing to mention is this idea of ROC, as I mentioned, the correspondence between the two. The data isn't as clear so far, but overall there seems to be this trend of the ROC score getting higher as we move from abstract black and white to realistic, but there's more that we need to do in this. One of the things we want to do is disentangle when the child identifies it correctly versus incorrectly. Is the gaze different? We also want to match beyond just age. We want to match on other behavioral developmental outcomes like verbal IQ, right? So if the child has really, really strong verbal IQ, is their gaze a little bit different? So those are directions that we're heading in. What is the future? We want to now get out of the lab and go into the real world. So we're now discussing with a couple of companies to do eye tracking in the real world, right? So two companies we're working with now. Very, very nice device. What you can see is that it's a small set of glasses. There's a video camera in the front that records what they see. The eye tracker is embedded inside the lenses, and at the same time, there's a small little computer like this that records the movements, right? So this is what it looks like in the video. Here's an individual just walking through a grocery store. Their task is very, very simple. They have to find just a bottle of shampoo on the rack. And you can see here the blue, or the red dot, I should say, is a perfect overlap registration of where they're looking at the actual visual scene. So we now have real-time tracking of their gaze in the real world, right? So you can actually do this from an analysis standpoint as well. This analysis is quite good because then what you can do is say, okay, I want to track this object, and then the software tracks the entire video where that object is and will count every time the individual looks at it, right? So really, really nice, you can see here this heat map that shows you where the individual was looking at. So now you've got a system to track eye movements in the real world. Let's put this on kids with CVI and send them to school, in their classroom, right? See and get a sense of an understanding of what it's like when that clutter is really demanding. What's a good day look like? What's a bad day look like? And then we now have, again, continuing that objective data classification and quantification, I think is absolutely crucial. So I want to thank I want to thank, first of all of you for your patience and waiting through this presentation. Many individuals who I mentioned, CVI Now, CVI Scotland, and the National Eye Institute, I think are fantastic resources to follow up in terms of this work. And I just want to I just want to end with what I call sort of the CVI family. I was looking over this, just collecting pictures over time of all the kids that we've worked with in the families. This, of course, is not complete. I've fallen way behind, but these are also individuals who have given me permission to show their picture. Elke, unfortunately, is no longer with us, but lives with us in spirit. Logan just had a baby a couple of years ago. The mother and baby are doing well. This young man, CJ, who you all know, saw his brain. He was an early participant. And when he saw his brain, he said, you know what, I want to be an MRI technician. And last year, he graduated from the Mass College of Pharmacy, now works for Shields MRI and Tufts Medical School, and then ran the Boston Marathon because he obviously didn't have enough time, things to do. Who else do we have here? We have Sebastian, we have Kyle. Oh yeah, we got Tina, because otherwise she'd beat me up if I didn't show her picture. Yeah, for sure. Yashai as well, who's pursuing his Ph.D. at Cambridge University. You know, the theory for everything. He's writing the theory for everyone. So it's really a wonderful approach. So I'm going to end it there. Thank you so